All right, so this is the second session of our comprehensive review, and we're going to cover DNA and RNA today. And so I'm sure you guys have heard of DNA before, but you'll have to know DNA very, very well for the MCAT. And let's just dive in. So the structure of DNA, like we talked about last time, we have amino acids, which are our monomers that build up our polymers, which are the proteins, right? Here, DNA is our polymer, and these nucleotides are our monomers to build up the polymer that is DNA. And just like we saw variability in the amino acids, there's variability in these monomers as well, in these nucleotides. And the structure of them is a little bit more complex than we saw with the amino acids. So let's look at the structure for a second. So there's two different classes. There's purines and pyrimidines, and each of them have different substituents, which defines what kind of purine or pyrimidine they are. And on top of that, they have a little bit of variability in what they look like. Where is it? Here and here, maybe. Here, okay, perfect. So the template is you have this sugar in the middle, which is a ribose sugar if this is an OH, or a deoxyribose if this is an H. So we've heard of deoxyribonucleic acid and ribonucleic acid, which is RNA, right? So that's determined by this group here on the 2' carbon. Then we have this glycosidic bond to a nitrogenous base, which is one of these purines or pyrimidines. Then we have this 3' OH, which isn't modified, but is very important. And then we have this 5' group, which can connect to several different phosphates. If it's 3 phosphates, it's going to be a triphosphate. If it's 1 phosphate, it's a monophosphate. 2 is a diphosphate. So if you've heard of like ATP, that's 3 phosphates. ADP is 2 phosphates. So back to the DNA. We have these four different nucleosides. Is it sides or tides? Yes, nucleosides in our DNA, right? We have 4 different ones because the identity of the nitrogenous base changes. You need to know how the nitrogenous bases look, and also how they affect the function of the DNA. So how does that work? Basically, they connect differently. They pair differently. So we probably know that A and T link to each other, and C and G link to each other. So notice that in A and T, we have 2 hydrogen bonds, and C and G, we have 3 hydrogen bonds. This has a big impact. The reason this has a big impact is because C and G bond pairing is stronger than A and T bond pairing. So how does this affect the properties of DNA? If it has more, we say more GC character. If the DNA has more GC bonding or more GC character, then it has a higher melting temperature because there's more bonding inside the DNA. So it's harder to denature GC DNA than it is to do AT DNA. So they could give you a question where they just are comparing different DNA strands. And if you notice that one strand has more GC, then you know it has a higher melting temperature. So the structure links to the properties. Okay. Any questions on this so far? Okay, that's another thing is memorizing these little substituents on the purines and pyrimidines. The way that I did it was I would have like little mnemonics. So the overall structure, the interior structure here is the same, right? It's a hexagon and a pentagon, and then these ends every so often. And then I would remember that this is here because adenine stands for amino above. So that's all A's. Adenine is amino above. So I put an amino above. And then for guanine, I would remember that for guanine, because it starts with GU, I would remember that an oxygen will go up So an oxygen goes up on top here. Then for cytosine, I would remember that the oxygen comes down. Rather than it being up here, it comes down and it swaps places with the amino group. And then the uracil and the thymine are basically the exact same, except for this methyl group. And methylation is going to become important throughout this. And so the way I would remember it is like uracil is the unmethylated one, whereas thymine is the methylated one. They're basically the exact same. So these, it looks very complicated to remember, but you just need to have a little bit of, memory tricks to remember how they bind. Another thing, so what else can we talk about here? Right. Okay, so now we talk about the phosphate backbone. The phosphate backbone is how, remember I talked about that 3' OH group, right? And I said that this is important. Why is this important? Because this is how these monomers link together. So we saw last time how amino acids link together. Here, this is the link together. So let's draw a little bit here. So this is our first nucleotide, and then this is our next one down here, and so on and so forth. Now the thing is, now we need to know what kind of linkage is this. This linkage right here is called a phosphodiester bond. So an ester group is like an R in the middle, an O and an O connecting to an R. Just a little bit of organic chemistry, right? So that's an ester group. This is a phosphoester because it's a phosphate in the center connecting to an O, an O, and an R, in this case, a carbon. Now, this is a diester because we actually have that connection going two ways. We have the R, the O, and the R going to up. And then we have another one, the R, the O, and the R connecting over here. So this is called a phosphodiester bond. And this is basically where we see phosphodiester bonds. And we talked about GC content. We talked about how it's more stable than AT. Chargaff's rules are basically just a math thing where it's like, since you have an A, then it has to connect to a T. If you have a T, it has to connect to an A. If you have a C, it has to connect to a G. There are exceptions to this, but they're not going to test you on exceptions. Basically, they can use this to ask you like math problems. So if it's like, if we have, right, so if we have DNA has a GC content of 30%, then you need to figure out that lets you know everything about the content of the DNA. Because if it's GC of 30, then it must be half guanine and half cytosine, right? And then the remainder has to be half adenine and half thymine. So if this is 30, then the remainder is 70. And if it's 70 between adenine and thymine, then it's going to be 35% each. And now you know the full content of the DNA just from this one fact. So those are Chargaff's rules. They can ask you like a very simple math question about that. Okay, now we get into the meat and the potatoes. Before we get into DNA synthesis, any questions on the basics of how they link together and these phosphodiester? Yeah, go ahead.
I think that MCAT ask questions regarding for the phosphodiester bond, like if you're making a new, I guess, bond. So I always get the directions confused. Is it like, oh, it's like 5', it's like 3' carbon? We can just go over that a little bit.
Perfect. We are going to talk about that in DNA synthesis right now.
Okay.
So perfect. So we are going to discuss DNA synthesis now, and DNA synthesis is very, very involved. And I honestly did not understand it until I studied for the MCAT, and I found this really, really nice video. So I'm going to walk through this video with you guys. So first things first, we have this directionality, right? So first of all, how do we have this numbering? The numbering is just like organic chemistry nomenclature. This is let's see here. So DNA is what we call anti-parallel. What that means is let's see. What that means is, notice how, so let's look at this. This is really important. So this over here, what? This part over here is what we were just talking about. This is the strand that's on its own that we were just looking at over here. Whoops. This is the strand on its own. This is a strand of DNA, right? But DNA is not a strand by itself. DNA is a double helix, two strands connected, and they don't connect in a parallel manner, but in an anti-parallel manner. You can see that one is right side up, so to speak, and the other is upside down, if we look at this way. So then the question is, of course, how do we actually like keep track of all this? What do we do to distinguish these guys? And the answer is that we need to mark it somehow. So the way we mark it is through the 5' and the 3'. So here we have the 5'. The 5' is over here, and we get that 5' from our marking of the nucleotide itself. 1', 2', 3', 4', 5'. So the 5' is this carbon that's attached directly to the phosphate, and the 3' is the OH group. You don't see the OH group here because it's attached to another phosphate. So the 3' hydroxyl, the OH group, attaches to the phosphate connected to the 5' end of the next nucleotide. So to show you that over here, we see that this is the 5' end, and this, look at how the last part on this strand is a 5' end. The top of this strand is a 5'. So therefore, this part is a 5' end, whereas we see that the top part here is a 3' end. The 5' is below it. So we would say that this strand, over here, that this end is the 5' end. Where's the apostrophe? This is the 5' end, whereas this is the 3' end. Oops. And then at the bottom of the strands, we would say that this is the 3' strand over here, the 3' end, and then this is the 5' end. So they run anti-parallel to each other. This one, I'm just going to arbitrarily draw an arrow going 3 to 5, and then this one, 3 to 5, is going this way instead. So this is what we mean by anti-parallel. So now you're going to see why this is very, very important in DNA replication, because the directionality of how we build DNA is critical. So any questions before I clear this and move on to synthesis? Okay, so let's go to synthesis. So I've just explained this 5' end and the 3' end, right? And now let's go to the synthesis itself. Okay, so semi-conservative model. I'm sure you guys have heard this before. What this means is there used to be like, debate over whether DNA was semi-conserved or not conserved at all. Meaning, does a DNA molecule make a brand new DNA molecule, or does some of the old DNA reside with some of the new DNA? And what we learned over time through a really great experiment, you're definitely going to learn about, is that it's a semi-conservative model. Half of the old DNA will pair up to half of the new DNA in both strands. So that's what we see here, where the old DNA is the blue and the new DNA is the green. And that's how DNA replicates, right? But then how does that actually happen? Let's see. Okay, so when we have DNA, right, we have two strands. Normally, they're all twisted up. When they're opened like this, and there's an origin of replication where the synthesis starts, and you can have multiple of these. And when that starts, over time, you start seeing the new DNA strands being formed. So here we have T1, we have at time 1, no DNA is being formed. At time 2, some new DNA is starting to form. At time 3, the DNA is getting larger and larger and larger. Until eventually, we have this happen. When these, red strands, which are the new strands, when they all get really long and they link up, right? When they link up, that's when we get time 4, which is where we have this brand new completed strand. And then we have 2 new strands of DNA that can twist back together, and now we have 2 new strands of DNA. So now how does that actually happen? We have to zoom in on a replication bubble and understand. And everything here you need to know is very critical. So first things first, we have the origin of replication, and these are still old strands, right? And now we need to talk about directionality. Yes. So if this is the 5' end, right, then the other side of it is going to be the 3' end, right? And so if this is the 5' end and this is the 3' end, on the other strand, on the complementary strand, right, it's going to be reversed because the 3 goes with the 5 and the 5 goes with the 3. So therefore, this is the 5' end and this is the 3' end. However, that's not it. Because if this is the 5' end going down this way, then over here is the 3' end at the origin of replication. And then if this is the 3' end here, then this is the 5' end. So notice what I'm doing here. This is continuously, this whole strand is continuously 5 to 3. Right? If this whole strand, if this end is 5, if the left side is 5, then the right side is 3. If the left side is 5, then the right side is 3. All right? And then the reverse is happening here. If the left side is 3, then the right side needs to be 5. And here, if the right side is 5, then the left side of that part needs to be 3. And so this is how the numbering works. And why this numbering is really important is because there's 2 different ways that we're going to be synthesizing DNA. So we've got our numbers here, right? And now we're going to look at how we actually create the DNA. So we have this enzyme called DNA polymerase 3. And DNA polymerase 3 is very important, or rather, it's important to know how the DNA how the polymerase synthesizes the DNA. It will read from 3 to 5. It reads from 3 to 5. And this makes sense because it's how we would normally read, right? We read from left to right, or we read from the small number to the big number or something, right? However you remember it, just remember that DNA polymerase 3 reads from 3 to 5. So we're going to be reading this strand 3 to 5. When I say reading the strand, I mean that the DNA polymerase will come here and it will look at the parent strand and it will say, okay, we're starting from 3 and we're building to 5, right? Now, when the enzyme goes down this line and builds, what is the directionality of the new strand going to be? Is it going to be 5 to 3 or is it going to be 3 to 5? Basically, 5 to 3, exactly. This is 5 to 3 here, this is 5 here and 3 here. So we need to keep in mind that we read 3 to 5, but we build 5 to 3 because DNA is anti-parallel. So it rides 3 to 5 on the parental strand and then builds 5 to 3. And that's, we can call the daughter strand, sure. And this is also the leading strand because it continuously makes the strand. It just keeps reading and it builds that strand. Now we have to look at the other side. Now in the other side, let's just do this before. So in the other side, let's say we have our DNA polymerase 3, right? And our DNA polymerase 3 reads in what direction? It reads from 3 to 5, correct? So let's say we start here and we build, right? And we build, right? And here's our other origin of replication. We read 3 to 5 and we build 5 to 3, right? What's the problem here? The problem is we are now stuck here, right? And what about this area? We can't, we started here, but the rest of the DNA over here isn't getting synthesized, right? Like this guy is gonna just keep going down that way. So he's fine, but we started right here. So what happens to the rest of the DNA over there? Well, the answer is we need to start again over here and continue until we hit the point that we stopped at, or that we originally started at. And so on and so forth. We need to keep going backwards and building until we hit this way. Why can we not just do this? Because DNA polymerase 3 reads 3 to 5. It does not read 5 to 3. So we have multiple strands on this end. And that's why this is called the lagging strand from another origin of replication bubble from behind. No, sort of. It'll make sense more here because he does it with smaller strands. So you'll see here. So here we have a small strand, and then we'll go further back in this. We're still in this origin of replication bubble, and we'll make a small strand, and then go back and make a small strand, and go back and make a small strand, like that. So it won't be to another origin of replication bubble. The origin of replication bubbles will just meet up eventually. So if we draw another origin of replication bubble, and if there's another bubble over here, this process will eventually, just because over here, right, there will be some red strands going this way. And eventually they will link up. Does that make sense? Okay. So, oh, actually, I didn't need to delete that. That's fine, I guess. Okay. So that's the difference between the 3 to 5 and the 5 to 3. And now I think we talk about primase. Oh, whoa, no. Oh, okay. I see. This is actually okay. This is good to know. Hold on. If this is, see, this numbering is hard to keep straight. So if this is 3 to 5 and this is 3 to 5, then this is also 3 to 5. Yes, this is also 3 to 5. So actually that one won't be lagging, it'll be leading. Which is to say, because this is another origin of replication. So here we're building continuously 3 to 5. Leading is always 3 to 5, yeah, because 3 to 5 in the parent strand and 5 to 3 in the building strand. It's leading because it's just going continuously, and the lagging one is slower because it needs to keep hopping backwards. So, and then we see on this side, this one is continuous here, but then over here is going to be the lagging strand. Make sense? Good. Yes. This is very hard to understand if you don't have like a total visual of it. It's very hard to understand through words. Like you have to see it. You just have to see it. So now, and these are called Okazaki fragments. You probably heard of them. These little fragments on the lagging strand are called Okazaki fragments because the guy who discovered it was named Okazaki. And that's part of the lagging strand. So we see that the parental strand, the one where the DNA polymerase is just writing straight, he's just reading 3 to 5 and he's just building continuously, has no fragments, right? But here he can only read 3 to 5 a little bit at a time before he needs to go back. Okay, I read 3 to 5. Okay, I hit the next strand, so I need to go back. Read 3 to 5. Oh, I hit the next strand, go back. 3 to 5. Oh, I hit the next strand, 3 to 5. This one doesn't have that problem. I read 3 to 5, 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 I read 3 to, and I keep reading 3 to 5. That's the thing. Until he keeps reading 3 to 5 until what happens? He keeps reading 3 to 5 until he hits the origin of replication. And so the little pieces are the Okazaki fragment, yes. Okay, yeah, and so he's just shown that this little thing is riding along the parental strand, making this. Okay, primase. Okay, primase is very important. Yeah, question. The strand direction, sure. Let's see. The strand directions. So the strand directions are this way. The DNA polymerase will read the parental strand, the line in black, from 3 to 5. He's reading 3 to 5. We are always anti-parallel, never parallel. So if we're reading 3 to 5, then we are building 5 to 3. Okay, so that's the leading strand. Now let's look at the lagging strand. The lagging strand is 3 to 5, right? But, or 5 to 3. Actually, I guess it depends which way you're reading, right? If we're just reading it left to right, then this is 5 to 3, but the polymerase starts at the origin of replication and runs along at 3 to 5. Or actually, okay, I get it. Leading appears to be pointed in 5 to 3 in red, but we said 3 to 5 before. Leading appears to be pointed 5 to 3 in red. 5 to 3 in red. So, okay, so the thing, when you say leading, right? The leading strand it's been built, it is 5 to 3 because it's being built 5 to 3. But it's the template that it's working on is 3 to 5. So this is, so yes, you have to be careful when you think about the leading strand and the strand, the parental strand that it's working on. The parental strand, so if we just write it out straight, if we just draw it out straight, if I have a parental strand here, if I have a parental strand that's 5, 3, then my polymerase is going to build starting from here in this direction because he reads 3 to 5, but this end is 5 and this end is 3 on my new leading strand. Does that make sense? Okay, good. So, okay, so now we need to talk about what enzymes are actually doing all of this, right? We have the concept, but we need to talk about the enzymes. So first things first, you'll notice there are some little green bits at the beginning of each strand. That's because, you know, how does the polymerase know where to start doing its work? It doesn't, right? It needs something to be there to tell it, like, the polymerase needs a substrate to bind onto, right? And the polymerase recognizes what we call a primer. And the primer is actually RNA. It's not DNA. It's actually RNA. And the polymerase recognizes the RNA and he's like, oh, okay, I can build here. And he starts building. He attaches there and he starts running along. This RNA primer is put down by an enzyme called primase, which is an RNA polymerase, right? Polymerase just means that it can build polymers out of these nucleotides. If it's a DNA polymerase, it uses DNA nucleotides, the adenine, guanine, cytosine, thymine, right? And it builds DNA. If it's an RNA polymerase, it builds RNA using adenosine, uracil, cytosine, and guanine. So this little green bit is made by primase, and then that allows the DNA polymerase to recognize the primase and start running along. So every single one of these Okazaki fragments has a small little primase on it before it starts. Whoop. Oh, actually, that's better. And then we have, so then how did this bubble even come about? Normally the DNA is wound up and twisted, right? There is an enzyme called helicase, which will unzip, it will unzip the DNA, and it will make it into this kind of loose, openish bubble that allows all these enzymes to come in and do the work that they need to do. So now, when we have DNA helicase opening, it unwinds or unzips the DNA, yes. Now, when we have this happening, okay, yeah, there's another good thing to know. So what about the RNA that we started with, right? Do we just leave it there? No, because it has to be DNA, right? It's not a DNA RNA mix, it's just straight DNA. So once the primer has allowed the polymerase to find it and go through, the primer needs to be replaced. Because it was RNA, it needs to be replaced with DNA. DNA polymerase 3 is in charge of building the whole of the strand. DNA polymerase 1 is in charge of replacing those primers. Those RNA primers are going to be replaced with DNA by DNA polymerase 1. Places RNA with DNA. And now lastly, we have topoisomerase. So now I have a really nice video that explains this. So this is essentially what DNA looks like, right? If it's twisted up. Well, this is more like prokaryotic DNA, which is like circular. Our DNA is linear, but the concept is the same. When we open up a replication bubble, right? This is what happens. Look at the twisting occurring. Look at the twisting occurring. It's just a physics thing where if you open up a bubble in this kind of like helix situation, you get this kind of twisting. You get this tension in the rest of the DNA. So how do we solve this? We have an enzyme called topoisomerase that makes small little cuts in the DNA, and that allows it to relieve the tension. It prevents it from twisting all up and getting all messed up. So that's the point of topoisomerase. And it prevents that what's called supercoiling downstream from the replication bubble. And lastly, we need the ligase. When does he talk about ligase? DNA ligase somewhere. Oh yeah, he's talking about right here. So the DNA, the leading strand, continuously goes, right? No problems there. But the lagging strand has these little bits, right? Where, we just have these gaps because the DNA polymerase had to stop and get off, right? And obviously that's not a full strand if there's those little gaps. So we have another enzyme called DNA ligase, which will come in and connect those. DNA ligase will only work on the lagging strand and connect those things, connect those Okazaki fragments together. And that's how we will eventually get a brand new, that's how we duplicated our DNA. So if we did all of this, right, eventually we'll go back to this situation. So essentially, that's what was happening in these small little bubbles. These small little unassuming bubbles, there was a hugely complex machinery at work that led us to 2 strands of DNA. So that is really big, really important. Any questions on DNA synthesis? Just find a good spot. This is a good picture, I think. Oh, it doesn't have helicase. There. If you wanna take like a screenshot or something, this is a good oh, let me actually give you the link to the video because the video itself is also very good. I just sped through it. Okay. Alrighty, are we good on this? Alrighty, cool. So now, good, awesome. Thank you. So now when we look at these diagrams, now we actually understand what the heck they're talking about, right? Because we have this RNA primer, we have our DNA polymerase 3, which is synthesizing DNA it says synthesizes in the 5 to 3 direction. That's correct. Builds in the 5 to 3 direction. Why? Because it reads in the 3 to 5 direction. Okay? And then we have this other strand, right? On the other strand, we are still synthesizing 5 to 3 because we're reading 3 to 5. Let me just, because right here is 3. Right there is 3. So this is being built. Whoa. So this the template strand is the parent strand is 3 to 5, and we synthesize 5 to 3, but then we get stuck at the end here. So that's the lagging strand. Okay. So we talked about the semi-conservative semi-conservative manner of DNA, and then we're going to talk about the repair. So repair is kind of weird because there's a lot of different like elements to repair, and it's kind of unclear like how much of it you're supposed to know. But I think the major things to know are the fact that DNA polymerase so obviously like this is a hugely involved process and mistakes happen, right? We do sometimes accidentally put like an A to a C or something like that, right? If there's a mistake like that, the DNA polymerase can usually figure out that it made a mistake and it will just replace it. So for example, for example, look right here, right? We put a T to a G. That's a mistake, right? The DNA polymerase will immediately recognize that and just cut it off, right? If it cuts it off, and this is called exonuclease activity because it's working at the end of the strand. That's what exo means. If it was working in the middle of the strand like this one, this is called endonuclease activity. Nuclease, enzyme that works on cleaves nuclear material, right? Nuclease. So this is so while we're replicating, we can do this proofreading activity from the DNA polymerase, right? And it can just fix things while it's going, and it recognizes it. And also very interestingly, this is normally our DNA is reading 3 to 5 and building 5 to 3, right? But in this proofreading, it can actually cut. It can reverse. It can back up. It's like a truck backing up and cutting it off. So it was normally building 5 to 3, but it can cut 3 to 5. Does that make sense? It's literally just backing up one. And this is, I believe this is mismatch repair. I think. So basically when there is, yes, so there's a variety of different repairs that you can do after the DNA is made. If the DNA was made and then there's a mistake, then we have a couple of different options, right? We have mismatch repair, which is like when we see, oh, this is messed up, it just takes the whole chunk out and it replaces it and we're good to go. That's mismatch repair. Then we also have something called when there's a thymine dimer. This is linked to UV radiation. When we have a thymine dimer, we can do nucleotide excision repair. It's basically the same thing, but it's specifically for these thymine dimers. then this is base excision repair. And base excision repair is, instead of taking out that whole chunk, we're swapping the single nucleotide. And of course, at the end of all these, DNA ligase needs to connect the new thing. Non-homologous end joining. I think this is more on the chromosome line rather than the DNA. Yeah, I think we talk about this when we review chromosomes. I think it'll make more sense then. But that's basically DNA repair. We have a lot of techniques to solve DNA synthesis is very good, but it's not perfect, and we have a lot of ways to address it if it goes wrong. Okay, so now we're going to move on to the central dogma. So the central dogma is probably the very first thing that your bio professor ever told you, and you probably didn't understand it then. You might not even understand it now, but basically, the DNA is, let's think of it like you're trying to cook something, right? So, and you're trying a new recipe that you haven't tried before. So what are you going to do? Let's say you get on your computer and you find a recipe that you like, right? So the DNA is like your computer. It's your store of all the information that you have. And you find the recipe that you want, right? And now you print out that particular recipe. And that's the RNA. You transcribed it from your computer to a piece of paper. And that's your RNA. And then with your RNA, so this is your computer where you found the recipe, and you print it out onto a piece of paper, which is the recipe printed out. And now you're going to follow the directions on that recipe, right? You're going to follow recipe, and hopefully you're going to produce, like, whatever food that you wanted. This is what happens inside of our cells on a daily basis, all the time, probably right now. So, but just as like a big example, that's the relationship between DNA leading to RNA leading to protein. Because this has to happen because our DNA is sequestered and stuck in our nucleus, right? It's not leaving willy-nilly. What's leaving is our RNA. So let's talk about transcription. Transcription. Maybe we go to RNA. Yeah, I think we can talk about this. Yeah, I think we can talk about this at the same time. So let's talk a little bit. We mentioned RNA. We've been talking about RNA, but what is RNA? RNA is, we know about DNA, and we know about DNA, and now we hear about RNA. So the major difference between DNA and RNA is that they're very, very similar, but it's missing this OH group at the 3'. Here in DNA, we have this OH, and in RNA, we don't. This is the major distinguisher, right? You should be able to look, by looking at these pictures, you should be able to identify, oh, this is RNA and this is DNA, just because of the 3' OH. Now, the other thing that sets apart RNA is that, well, there's two other things. The first thing is that we have this uracil nucleic acid, uracil. And the uracil, I think we showed earlier, the uracil is the unmethylated version of thymine. So we have ATCG for DNA, and we have AUCG for RNA. So that basically they're just showing, this is a nice diagram. Basically, thymine is only found in DNA and uracil is only found in RNA. So now, differences, right? The other major difference is RNA is single-stranded. RNA does not form this double helix like DNA does. So why is that, right? Why is that? That's a very, very important thing about why. DNA is, the point of DNA is that it needs to retain all of our genetic knowledge, all of our genetic information in a very stable, in a very, locked up tight way. That's why we have this very stable double helix that is very protected and locked up in our nucleus. And also you'll notice, we talked about last time with the proteins, right? This sugar phosphate backbone, what kind of properties does it have? The properties that it has is that these oxygens are very negative. Remember when we talked about proteins, do negative charges want to be on the outside or on the inside of a protein? That's a question I'm asking. Something related to polar, yes. Is negative polar or not polar? Negatives are polar. Negatives are polar. Charges are polar. Is water polar or non-polar? Water is polar, right? So will polar want to be with polar or will non-polar want to be with polar? Polar with polar, correct. So if we have our water over here, does the water want to be next to the negative backbone or on the inside with these aromatic groups? Next to the negative, exactly. So now we start to understand why DNA is the way it is. DNA is set up in such a way that the negatives are exposed to the aqueous environment where it's the most stable. And the aromatic groups, the nonpolar groups here, like these, these big chunky carbon rings, these guys are hidden away and stabilized to each other with their hydrogen bonds. So this is all set up very, very well. These guys are negatively interacting with all the, all the water out here, and they're stabilized. Whereas these groups are inside and they're stabilized. And so why does this all matter? Because the structure relates to the function, always. The structure of DNA is that it's locked up tight and it's not meant to be accessed. It's meant to be stable. Whereas the structure of RNA is much more free because it's meant to be accessed. It's meant to be read. It's meant to be used because it's just temporary. It's just a transmission of the information. If I have the recipe and I spill, some egg on it and I can't see it, is that a big deal? No, I can just go print another recipe. But if I spill some water on my computer, that is a big deal. I can no longer access the source of the information. So does it make sense the difference in function? One is for stability and for information retrieval. One is for information transmission, and the stability doesn't matter as much. It's the accessibility that's more important. So the RNA is just meant to be temporary and to pass on information. And you can see it in the structure of these two molecules. Yeah, that's basically what they're saying. Advantageous for the cell to minimize damage and degradation DNA suffers. DNA needs to be stable. Double-stranded motif provides DNA the stability it needs. The single-stranded structure allows RNA to pass genetic information and be quickly recycled once it's completed its function. It is temporary. So we talked about DNA versus RNA. These are some of the major differences. We talked about uracil versus thymine. We talked about double-stranded versus single-stranded. We talked about deoxyribose versus ribose. Now here's the big deal. There's primarily one form of DNA, double-stranded DNA, right? It's called dsDNA, double-stranded DNA. There are multiple forms of RNA because there's multiple forms of information that we need. The first one is mRNA or messenger RNA. And this is why I went to hear from Central Dogma, because mRNA comes up in transcription. So what is transcription? Let's see. I don't like this picture. Oh, I like this picture. Okay, good. So So our cells are not making all of our proteins at the same time, right? That would be a waste of energy. We're only making the things that we need in response to the environment. So let's say that, there's a particular protein that we need for whatever reason in the cell, and the signaling from the environment just came into the cell that, hey, we should start making this protein. We need this protein, right? So we'll have the cell signaling pathway that's eventually going to lead to the production of something called a transcription factor. A transcription factor is very important because it's able to cross this boundary between the between the nucleus and everything outside of the nucleus and the cytoplasm, right? Because it basically is another messenger. It's a signaling molecule that comes in, directly binds to the DNA, and it will have some kind of effect on gene expression and protein production. It will either tell the DNA, hey, let's start making this protein, or it will say, let's stop making this protein and silence it. All right. Either way, it has an effect. How does it have an effect? Very important. So this is transcription. First things first, we have here, this is how our DNA is just chilling in our nucleus, right? Once we have transcription factors bound, it's going to attract an RNA polymerase to come attached to the promoter region of the DNA. Once the RNA polymerase attaches, this, you can think of it just in the same way as like the primer in the DNA synthesis, right? We have a primer, and then the polymerase comes and it starts running along. Here we have a transcription factor, and the RNA polymerase will come and it'll start running along. This is RNA polymerase versus DNA polymerase, right? So the RNA polymerase is now going to create a strand of RNA. And this is again another thing with directionality. It's very interesting. If I am creating a 5-3, right? I'm creating a 5-3 mRNA strand by reading the 3-5 DNA strand, correct? Right? That's so far, I think we covered. I want to draw. This one is 5. This end of the RNA strand is 5, and this end is a 3, right? So if we are building 5-3, then the regular strand itself must be 3-5, right? Now here's an interesting question. What is this actually coding? What is this RNA actually copying? Is it copying this strand, the one that it's attached to, or is it copying this strand up top? And you might be able to see from the picture here that this is called the coding strand because we're making a copy of this. We're making a copy of the top one. Because DNA is anti-parallel, we attach to the one that's complementary to the one that we actually want. If we attach to this one, then we would end up making this DNA. But if we want this DNA, then we attach to the partner, to the complementary strand, and then we make a copy of it. Does that make sense, or is that confusing? So here we have two different parent strands, right? We've got this strand. Let's call this the red strand and the blue strand. No, actually colors are going to be confusing because everything's colored anyway. Let's call this strand number 1 and strand number 2. I'm only talking about the parent DNA right now. And then we come in with this polymerase and we want to make a copy of strand number 1. To make a copy of strand number 1, we don't attach to strand number 1. We attach to strand number 2 because strand number 2 is complementary to strand number 1. By attaching to strand number 2, we make a copy of strand number 1 because this and this are the same thing. Almost. This strand right here, this strand, and this strand are actually parallel. This strand and this strand are actually parallel. Whereas the blue strand is anti-parallel to both of them at the bottom. Does that make sense? Why do you want to make a copy of strand number 1 again? Okay, very good. The reason that we want to make a copy of this DNA is because the mRNA is going to, the DNA cannot leave the nucleus, but the mRNA can leave the nucleus. And when the mRNA leaves the nucleus, it can get turned into proteins. This is why we want to make a copy of the DNA. The DNA is like our computer, right? The DNA is our computer that we have the information on, and we need to get the recipe from the computer. So the recipe is this mRNA strand that we're making, and then the mRNA strand can come out to the kitchen, the cytoplasm, and we can make our proteins. Is this also semi-conservative? No, because the DNA is not involved in leaving the nucleus. It's only the mRNA, the brand new mRNA that we just made, that is leaving the nucleus. So semi-conservative specifically means that we made that we started with this red DNA, and then once we make once it duplicates, we end up the 2 parent strands are going to be attached, to 2 new strands. Why is it doing this? To 2 new strands. Oh, whoa, why can I not draw here? What's going on? Okay, there we go. To 2 new strands. And this is what we call semi-conservative. It would be conservative if it would just made purely new red strands. That means that there's no new material. It's all the original material. But in this case, it's conservative. The reason that mRNA is not semi-conservative is because mRNA is simply putting out 1 brand new strand. Does that make sense? Okay, perfect. Any other questions? Alrighty. Okay, so let's keep going. So we have created this mRNA strand, right? Before it leaves the nucleus, there's some things that we need to work on the mRNA strand to make it ready for translation. And this is specifically in eukaryotes. In prokaryotes, they don't care. They just ship it off. In eukaryotes, because we have that separation between because in prokaryotes, they just have a nucleoid region, right? They don't have a nucleus. They don't have that boundary. We have that boundary. So we are very careful about what we let go and what we let come in. So what we do is so now we've created our mRNA, right? And this is a copy of the DNA of one of the DNA strands, right? Actually, just from these numbers, you can tell exactly what was copied and what wasn't because of the numbers. And this is important. This can be talked about. This is a 5-3, right? So this is a copy of the top one, right? Which is the 5-3. That means that the polymerase was building based on this 3-5 at the bottom, because by building on the 3-5, it made a 5-3 here, and that's what we have here. So we built upon this one, which was anti-parallel to the copy of the thing that we wanted. That's essentially, so even from the numbers, that's why the directionality is so important. From the numbers, you get a lot of information about what's happening if you understand how it works. So now processing, post-transcriptional modifications. We have this, what we call pre-mRNA. You probably heard of exons and introns before, and it used to confuse me so much. We do this thing called splicing that removes some parts of the genes that are not involved in coding, right? What I finally learned that allowed me to remember this is that introns stay in the nucleus and exons exit the nucleus. I wish somebody had told me that, but I didn't. But I found that out later, and I was like, "Oh, okay, that makes a lot more sense." Because I would always be so mad, like why the introns wouldn't stay in the DNA and or the RNA, but now I was like, "Oh, okay, I guess that makes sense." So introns stay in the nucleus. Remember, right now we're still in the nucleus. All of this stuff that we just talked about is still happening in the nucleus. So the introns are going to be cut out. And we've got these exons. And then we're going to do two other things. We're going to attach this 5' methyl guanosine cap. It's just something that we attach to the beginning that's going to help this thing to be recognized for translation. And we attach something called a poly A tail, which is a long string of adenines rather. Now we have mature mRNA. This thing is ready to be translated. So now we leave the nucleus finally. There's also, this is basically the same thing about transcription factors, right? If it's an enhancing transcription factor, it's going to cause the mRNA to be made. If it's a silencing transcription factor, it's going to stop the mRNA from being made. And that's going to lead to either more protein production or less protein production. So what do we do with our mature mRNA? Now, so if we look back at the central dogma, right, we just made our mRNA. That's the middle step. Now we need to translate it into a protein. So after building the mRNA, it separates from the anticodon. Yes. Wait, what do you mean by anticodon? What do you mean? You mean this part?
Yes, yeah.
The antisense DNA strand.
Yeah.
Yes, it separates and the DNA is going to go back to being its normal, helix self.
Okay, thank you.
You're welcome. Yes. So, it's going to leave like the mRNA was never there. It's just copying and then it's getting out of there and it's going to the cytosol. So now, oh, I don't like this picture though. I had a better picture of this. Where did I have it? Oh, yeah. Oh, also, this is like a fun fact to know for some reason. I have no idea. I don't know. They just want you to know this for some reason. So there's two different kinds of ribosomes. In prokaryotes, we have 70S ribosomes. Eukaryotes, we have 80S ribosomes. And so I just remember it's 30, 50, 70, 40, 60, 80. It's by 20 each time. So that's just a pattern. And I remember that the prokaryotes have less because, you can think of them as like lesser life forms compared to us eukaryotes or something. So translation. Yes, I had the good pictures here. Okay, so, all right. So we made our mRNA and our mRNA needs to get turned into protein. So how does that happen? Well, we have these little guys called ribosomes. And ribosomes' jobs are to build proteins. And that's why ribosomes are actually the only organelles that prokaryotes have, because they need proteins too. So, this is how ribosomes are made, which is kind of cool too. It's the same thing, right? But it's rRNA. Before it was messenger RNA, now this is ribosomal RNA, right? So we have this ribosome stuff and it comes out and then these subunits are going to pair up and then we have a ribosome. So what is a ribosome going to do for us? When the mRNA comes, it's going to find these ribosomes are going to get attached at the 5' cap, and it's going to scan along the mRNA until it finds the start codon, AUG. So what are codons? Actually, I need to talk about codons first. What is the right way to talk about this? Codons. Oh, the triplet code, yes. Yes, okay. So how do we get from DNA and RNA to a protein, right? We have a genetic code, essentially, right? We have these each nucleotide is adenine, a uracil, guanine, adenine, uracil, cytosine, et cetera, et cetera. 3 of these make up a codon, and a codon translates directly into an amino acid, and amino acid chains are proteins. So this is how we do that. And we have a whole, a dictionary. You don't need to memorize, oh, this means this. But there are some that you need to know. AUG codes for methionine, and it's the start codon. The ribosome is going to look for the AUG before it starts actually building the protein. So AUG is a start codon, and I think the mnemonic for this is that school starts in August, so that's why it's AUG. Then we have 3 different stop codons: UAA, UAG, and UGA. And Kaplan has a great mnemonic for these. I'm going to put it in the chat. Yeah. So those are the stop codons. Once the ribosome reads these, it will stop the protein production, and that's it. It's over. And they don't code for an amino acid. They're just like, okay, let's stop. We're not going to do it. We're not going to do anymore. But it's very important to know that the first amino acid is always going to be a methionine. So codons. So I've explained codons, and now, yes. So remember we talked about anti-parallel, right? So this shows up again. If AUG is on the mRNA, then we have this. So now we have, so we have 2 different types of RNA happening here. Actually, 3 different types. We have the rRNA in our ribosomes. We have the mRNA carrying our recipe that needs to be translated. And then we have something called tRNA. And tRNA is transfer RNA, I believe, right? tRNA. Is it transfer RNA or is it? It should be transfer RNA. Yeah, it's transfer RNA. Okay. So, where are the pictures? Where are the pictures? Here. So, okay. So this is a little bit complicated. This is again another thing like this is another type of synthesis, right? So once the tRNA, there's 3 different sites in the ribosome, and the tRNA attaches in the P site. And then we have a cycle happening that builds up our polypeptide chain. There's an A site, a P site, and an E site. You can remember it as like the arrival site, the polypeptide site, and the exit site. So what's going to happen? This codon, right? We look at the mRNA here. The AUG has been read, and now we're building here. Then a tRNA with the anticodon, it needs to be anti-parallel to whatever the codon is here. It's complementary, right? It's anti-parallel. It comes in and attaches here. Because it attached properly, we're going to have the polypeptide chain attached over here. And then the whole ribosome is going to shift to the right. Once it shifts to the right, what happens is this guy now moves to the P site. And the empty tRNA, which no longer has a chain, it moves to the E site and it exits. And this continues onwards and onwards and onwards. And this polypeptide chain gets longer and longer and longer as the mRNA continues to extend to the left. Now, how does this end? The stop codon is UGA or UAA or UAG, right? Once there is a stop codon, something called a release factor will bind to the stop codon. Once that happens, it's over. We're done. Everything is going to separate. And now we have made a protein right there. That's our protein. And after that, all of this stuff can just get recycled again. The RNA, the nucleotides, will get recycled. The ribosomes will go find another mRNA to make proteins out of. The tRNAs will go and get charged again, and they will pick up another. By charge here, I just mean that they will go pick up another amino acid to be reused. So it's very economical. Yeah, so this is like an overall picture of everything we talked about. Inside the nucleus, we've got our DNA just chilling. And then we have, once there's a transcription factor saying, hey, RNA polymerase, go to the DNA and get me this particular recipe. So it will make that particular recipe, right? And then still in the nucleus, we're going to do a little bit of processing to make it ready for translation. So we transcribe this pre-mRNA, we attach the 5' cap, we get rid of the intron, and we attach the poly A tail. And that gives us this lovely mature mRNA, which is now allowed to leave the nucleus. And then we have translation. The ribosome comes, finds the 5' cap, attaches, starts translating. Once it hits the stop codon, it stops, everything disassembles, and we have a completed protein. And this is basically what you need to know about the central dogma. This is the central dogma. We start with DNA, we transcribe, we get RNA, we translate, and we get protein. Any questions about the whole central dogma? Okay. Good. So now let's see. Yes, so we were on transcription and translation. So we covered transcription, we covered translation, we covered codons, degeneracy. That's a little interesting thing. We can talk about degeneracy. Codon degeneracy. So codon degeneracy is the idea that multiple codons can lead to the same amino acid. And this is good because if these codons were really, really specific, then we would have more mutations. So if all three of these led to something different, then we would have more mutations occurring. But because we have this kind of flexibility in the third one, then we have less mutations occurring because more combinations lead to the same amino acid. If they were very, very specific, then we wouldn't have as much wiggle room for mistakes. But in this case, we do have wiggle room for mistakes. You can see here, the CUC is, coding for a leucine, but the CUU is also coding for a leucine, which is good because if we want a leucine, then making this one little mistake here shouldn't screw up our whole polypeptide process. And so this is called the wobble hypothesis. I like to think of it as like the wiggle hypothesis because it gives us a little bit of wiggle room to make mistakes. And this is specifically about the third nucleotide. The third nucleotide is not having that much effect on what we are making. There's one exception, of course. And the one exception is, of course, if we screw up on AU, right? Why is that? Because if we screw up on AU and we put in a G when we meant to put in one of the other three. Oh, no, sorry. Wait, I'm messing up. Well, actually, no, this can also be a problem, right? Because we could accidentally start when we didn't mean to start yet, right? So that's one problem. The other problem is if we have a UA and we mean to make a tyrosine, or a UAU or a UAC, and we mean to make a tyrosine, but accidentally stop, right? That's really bad because now we have a non-functional protein because it stopped probably earlier than it was supposed to. So actually, I don't think we talked about mutations, right? Did we talk about mutations? Oh, it's right here. Mutations. Yeah. Okay. Mutations. Yes. Okay. So there's a few different types of mutations. If the mutation doesn't change anything, and it's in that third spot where we have wiggle room, it's a silent mutation. So it's not a problem, right? We still get what we wanted. But if we lead to an error, if we meant to do like glycine, but instead we put in a cytosine, that's no, cytosine is not a amino acid. If we meant to put a glycine and instead we put like a tyrosine, that's called a missense mutation because we made a mistake that had an effect on the amino acid. And finally, we have a nonsense mutation. And a nonsense mutation is what I was talking about by accidentally putting up the stop codon early. If we accidentally stop it, then we have a nonsense protein now that doesn't work. Yes. So I think those are the major things. We talked about transcription factors, and that leads to gene expression or gene repression. Nuclear localization. So nuclear localization sequence is basically just the thing that transcription factors have that allow them, it's like a passport. It allows them to get into the nucleus. Remember, we're very careful about, what we allow into our nucleus because this is our all of our knowledge, right? So we don't want to let anything in here. We want to only let very specific things that are supposed to be in here. So the nuclear localization sequence localizes or moves the transcription factor to where it needs to be, the nucleus. And activators and repressors. Okay, this is just another layer, right? We have transcription factors, but if there's a repressor binding it, then the transcription factors won't have an effect. And if we have an activator, then it's going to encourage transcription factors and polymerases to come and start transcribing. So basically, there's just various different ways to regulate our gene expression. Transcription factors are these things that what are they specifically? Good question. What are they specifically? Transcription factors. Eukaryotes require several other proteins called transcription factors to first bind to the promoter region and then recruit the appropriate polymerase. So I guess they're just other proteins, and their job is to make the transcription happen. And they have that nuclear localization sequence that that gets them to where they need to be with the DNA. And as we saw with the as we saw with this image, these transcription factors are going to be created as a result of outside signaling, right? Our cells are very economical with our resources, and they only make things based on the environment. And well, not necessarily only, but they make things in response to the environment. And actually, a really good example of that is this. So this is an example of an operon. And an operon, this is like a model of how prokaryotes are transcribing, are controlling and regulating their transcription. And so how this works is, this is a lac operon. And if there is no lactose present in the environment, it creates this repressor protein that binds to the operator zone. The operator is just like a region that controls what should we or should we not utilize these genes and make proteins from them. So the RNA polymerase is not allowed to make these proteins and genes if there is no lactose present. So let's think about this, right? There is no lactose present. So do we need enzymes that break down lactose? If we have no lactose in the environment, do we need to have enzymes that break down lactose? No, right? So if we don't need them, then don't make them. So if we don't want to make them, there's a system well, not our, because this is a prokaryote, but there's a system to not make them. The system is, in this case, the system is we make a repressor as long as there is no lactose. Therefore, the repressor prevents any lactose breakdown enzyme production. And now we don't have any unnecessary enzymes because we don't need them. So why would we make them? Now, what if there starts to be lactose present in our environment? What if they run into a situation where there's a lot of lactose? Obviously, if there's a lot of lactose, then we probably want to start breaking down that lactose and using it, right? So now let's say that there is lactose present. Do we need those breakdown enzymes? Yes, we need those breakdown enzymes because we want to utilize that resource. So yes, so let's make those enzymes. So how do we do that? And what you can see here on the screen is when there's lactose present, the lactose binds to the repressor. Do you see the little yellow lactose molecules are binding to the repressor? Presence of lactose inactivates the repressor. And since there is no repressor activity, that allows for the transcription of the breakdown enzymes. Therefore, we now have the ability to break down the lactose. So this is a very elegant way to only produce what we need to produce based on the environment, right? Instead of wasting energy making all these enzymes all the time, and if there's no lactose, then we don't use them. That's a waste. Instead, we have this kind of repressor that's very, very like clever. If there's no lactose, it shuts it off. If there is lactose, then it allows it to run. And so this is an example of gene regulation in prokaryotes through operons. So even prokaryotes are very, very efficient and good at regulating their genes. So let's see. Yeah, they won the Nobel Prize for discovering this. Yeah. So any questions on this lac operon? This is it's a very I don't know, I find it cool. I think it's pretty cool. Make sense? All right, cool. Now, oh, just some vocabulary here. So polycistronic versus, is it monocistronic or is it not? Yeah, it is monocistronic. Okay, so prokaryotic genes, by polycistronic, one mRNA can make different proteins because there's different start sites, right? So we can make a protein here, a protein here, a protein here, and it really doesn't care. It can do this all at the same time. The prokaryotes don't care. Eukaryotes care a lot. They're like one mRNA for one protein, and that's it. We're not messing around. They are very, very clear about it. One mRNA for one protein, you don't get any more. Prokaryotes, they don't care. They'll do a bunch of proteins from one mRNA. That's just some vocab. Polycistronic versus monocistronic in eukaryotes. Okay, so what else? Methylation. Yes, that's an important thing to talk about. Oh, actually, oh yeah. Okay. Yes, so let me clear this. Exceptions. Yes, exceptions. So you've probably heard of retroviruses like HIV, and they use something called reverse transcriptase. So they have found a way to bypass the system. So basically, right, we want to maintain this integrity of the DNA. We don't want anything happening to our DNA. We only want to turn it into RNA sometimes to make protein, right? These nasty retroviruses have come in and said like, oh, you know what? I've got this RNA and I want to put it into your DNA. So you know what? I'm going to turn my RNA, my viral RNA, backwards into DNA, and I'll trick you. This is your DNA now. You get to have it, right? And then our cells get infected with this viral DNA because retroviruses found a way to, subvert the system and kind of sabotage it in a way. So we have this thing called reverse transcriptase, which is the reverse transcriptase creates a double-stranded DNA molecule out of the RNA that they possess, right? Is this a DNA polymerase or an RNA polymerase? I didn't fully explain what a DNA polymerase is and what an RNA polymerase is. But if it's making a DNA molecule, then it's a DNA polymerase. If it's making an RNA molecule, it's an RNA polymerase. So reverse transcriptases are actually DNA polymerases. So yes, violate. That's a very good way to explain it. Retroviruses violate the central dogma, and it's like, I don't know, it's almost like a crime like that against nature. But anyways, so instead they produce DNA using RNA. Oh yeah, they talk about HIV here. Once HIV infects a cell, the virus uses a reverse transcriptase to create a DNA molecule from its viral RNA. And then the virus uses machinery within the host cell's nucleus to splice newly synthesized viral DNA into the host cell genome. So it's kind of very scary. Like they have figured out a way to upload bad information into your genes permanently. So it's like if you have a computer and your computer has all these important documents on it, and somebody comes in with a flash drive and they plug it into your computer, and the flash drive screws up your important information. That's sort of what reverse transcription is. We're going backwards into the DNA. Any questions on retroviruses? There's a lot. I'm sure there's probably more about retroviruses I can talk about. Oh, this is it's later. It's with cell bio. Oh yeah, we'll get to that. Okay. Transcription, translation. Talked about this. Oh, this is important. This is important. Heterochromatin versus euchromatin. Okay, so wait, is this what I want to talk about? No. Well, it's sort of what I want to talk about, but the picture isn't good. Hetero. Yes, okay. This is more for I think we'll talk about this in meiosis stuff. Yes, this is important. Okay, so we talked a little bit about gene silencing and activation, right? We said like genes can make more proteins or less proteins based on the transcription factors and what's going on. But how does that actually happen? Oh, excuse me. How does that actually happen on a molecular basis? On a molecular basis, we talked a little bit about how the DNA has this negatively charged backbone, right? And so, yes, I think this is the best way to explain it. So, but first I need to explain histones. Okay, I can't explain this yet. I need to explain histones. Okay, I guess I can explain it this way. Yeah, I guess I can explain it this way. So DNA, as we know, we see it two different ways, right? We see it like, we see it, if we just search for DNA, right? Then we'll see, this long double helix, right? But in the cell, in the nucleus, right? We see it like this. We see this X shape, right? We see the chromosomes, right? And that is DNA, right? And then more than that, we also see this is kind of a good one. So we have this chromosome, and then we have chromatin, and then we have histones, nucleosomes, and DNA, right? So it's like, what is all this? Like, what's happening here? What's happening is our DNA is very, very tightly compacted together. Very extremely tightly compacted. If you unwound all of it, it would be like very, 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 very long. So what we do is we bundle it all up into this extremely tightly woven structure. And what the DNA is bound around is these things called histones, these proteins called histones. And so histones are these little, these little balls or beads, right? The DNA is negatively charged. What charge will the little histones protein have if you want this binding to be really tight and good? If the DNA is negatively charged, then the nucleosomes are positively charged. So the cluster of histones are a nucleosome. Now, when this is all bound, right, the DNA is inaccessible. We cannot transcribe off of this. It needs to be loosened for the DNA polymerase to access it and transcribe. So when it is very tightly bound like this, you can call it silenced DNA. And silenced DNA is called heterochromatin. It's closed. They are not expressing because they are bound up. They're tight. You can see heterochromatin here. You can see the DNA is very, very tightly bound to the histones. This is called heterochromatin. We are not transcribing these genes. These genes are off. These genes are off. They are not being used to make proteins because they're so tightly bound. This is in comparison to euchromatin. Euchromatin, which is loose. You can see it's still wrapped around the histones, but they're not all bundled together. There's a little bit of looseness here. And this is a spot for the RNA polymerase to work on and start making mRNA. So here, these genes are considered on. They are able to be transcribed. Now, what is the difference? How do we get heterochromatin versus how do we get euchromatin? The answer is through something called acetylation, histone acetylation. An acetyl group is this thing right here in purple, acetyl. Remember that we said the DNA backbone is negative. Therefore, the histones will be what charge? If the DNA backbone is negative, the histones are positive. Yes. Therefore, we get a positive negative bond, right? An ionic bond, which is very good, which is very strong. It allows it to bind tightly. If it binds tightly, we have less gene expression. Now, if we neutralize this positive, yeah, it's positive. If we neutralize this positive, will the attraction between the histone and the DNA increase or decrease? Decrease, yes. Awesome, everybody's on it today. So originally we had for heterochromatin, we have a strong binding ionic bond, right? Which is leading to less gene transcription because it's so wound up, right? And we can't access it with the DNA polymerase. But now in euchromatin, we have disrupted the bond with this acetyl group, which leads to more gene transcription because it's now more open. You see how it's a little more open now because this binding isn't as tight. So this is what we mean by acetylation. And so histone acetylation is going to activate a gene, right? Because it's preventing that strong binding. Now, let me see if we can do this card. Yes, okay, cool. So this is, so now if we do if histone acetylation is gene activation, right? Histone deacetylation, removing those acetyl groups and allowing the binding to happen, would lead to gene activation or deactivation. If acetylation is activation, right? Link the A with the A. Acetylation is activation. Deacetylation is deactivation, right? Exactly. Why is it deactivation? Because we have to think about that bond, right? We have to think about the bond. The bond is showing us if we get rid of this acetyl group, then we'll make this positive and negative and we'll bind tighter. Now, here's another thing. We have this thing called DNA methylation, which is addition of these small little methyl groups. Here is a question. methylation, should it increase or should it decrease this binding? Let's think about that. A methyl group, it's not positive or negatively charged, but just by its excuse me, just by its existence, it it has electrons. So it's slightly negative. No, this is a totally different concept from acetyls. This is totally separate from acetyls. So yeah, this is totally separate. I'm moving on to something slightly different. So the methyls are slightly negative. So if the DNA has this slightly negative, they're not negative, but they have electrons, right? So it's a slight polarity. If these methyls are added to DNA, do you think it would increase or decrease its binding? If methyls are electrons, do addition of electrons make DNA more or less negative? That's our first question. If we add methyls, is the DNA becoming more or less negative? More negative. Good. If our DNA is more negative, is it binding to the histones more or less? More, increases. Good. If the binding increases, then the genes will be activated or deactivated. Deactivated. Why? Because it's less open. The more it's binded to the histones, the less open it is. So notice, so for the new members here, notice how we are going through each point a bit at a time. We could jump from methyls, we could just jump and say methyls lead to gene deactivation. Yes, that's true, right? I could have just written, oh, you need to know methylation leads to gene deactivation, right? I could write that, that's true. But the reason I go through this one step at a time is because if you understand why it works, then it's much harder to forget it. Because if you're on a test and you just know this, and you're like, wait a minute, was it activation or deactivation? Wait a second, which one was it? I forgot, right? But if you know that methylation is electrons, then electron could be more negative. And if it's more negative, then it's probably binding more to the positive. And if it's binding more, then it's probably deactivation. That's how you should try to study like everything, because you can break down everything into these very, very small relationships. And these very small relationships, when you link them together, give you very, very powerful outcomes and explanations. Yeah. So good. So that is basically what you need to know about gene silencing and activation. And you can see how, how we talked about the relationships. Any questions on euchromatin and heterochromatin? I need to see. Oh, I need to close this. Epigenetics. So we talked a little bit about epigenetics with the so epigenetics just means like changes that happen to the DNA as a result of like, yeah, physical changes on DNA that amplify or silence the expression of certain genes. So what we talked about with the histone acetylation and methylation, that's a form of epigenetics because we're modifying the expression of the genes. Imprinting. Imprinting. I don't remember imprinting. What is imprinting? Imprinting. I've never heard of imprinting. That's why I don't remember it. Imprinting. Is that a thing? I haven't. Okay, I've never heard of that, so I don't know if that's a thing or not. Imprinting occurs when an inherited copy of a gene is silenced due to epigenetic modification passed on from parent to offspring. Inherited from biological father and mother. During gamete maturation. Interesting. Yeah, I don't know too much about this, so I can't speak on it. I don't know if this is important or not. I've literally never heard of it. There's a lot of this. There's a lot of stuff with like this bio stuff that like if you go too deep down, then you find things that are like not actually relevant to what you need to know. But it could help you understand it more, but it wouldn't be there. So we talked about histones. Talked about chromatin. The wrapped DNA is chromatin. Talked about acetylation, deacetylation. Talked about methylation and of course demethylation. X inactivation. Okay. Okay. So this is sort of this is more along with the lines of genetics. I guess this is genetics, but not exactly. Inactivated X chromosome. In females, embryonic cells randomly inactivate one of the X chromosome to become a body. Oh, right. So, yes. This is just a thing where like, this is kind of hard to explain. They basically inactivate the X because they don't want in females X. There's two X chromosomes, right? So instead, to prevent having too much expression of X-linked genes, then we use epigenetics to inactivate some of them, and we randomly silence either from the mom or for the dad. So becomes so heavily methylated is classified as heterochromatin, and that is just a way to prevent overexpression of some of the X-related genes. Cancer. Cancer. Yeah. Oncogenes. Yeah. We can talk about that. So okay. So this is pretty interesting because this is very much. So we have oncogenes, which are genes that are implicated in cancer, right? If those genes become mutated and are active, they are increasing cancer's activity. Then we have tumor suppressor genes that are genes that are against cancer. So they are genes that slow down the cell cycle and repair DNA and will trigger cell suicide if it sees that, the cell is beyond repair, right? Now, so here's an example. A proto-oncogene means that it is not, it could become cancerous if mutated, but it's not yet. So these are genes that are like we need normally for like the cell cycle, right? Like let's say they're genes involved in cell cycle, in running the cell cycle, right? And if there is a mutation in the proto-oncogene, in one of the alleles, it will activate it, and now our cell cycle machinery is probably going haywire, right? And that is cancerous, right? If we increase our cells' ability to grow and duplicate, that's leading to cancer. However, we have these helpful tumor suppressor genes, which are there to stop that, right? Any kind of nonsense with the cell cycle or growing too fast or anything like that, the tumor suppressor genes are going to try to take care of it. We get cancer if we have inactivating mutation in both tumor suppressor genes and just one activating mutation in a oncogene. So this situation is cancer because both of our safeguards have been inactivated via mutation, and one of the potentially bad genes has become bad, and now we have cancer. Now we have cancer. So this is related because here we have some silencing mutations. We're silencing the function of our guardians, and we have activated the function of a cell of a gene that should not be this active. So overall, you need 3 mutations for cancer: 2 inactivating on both copies of the tumor suppressor, 1 activating on 1 copy of a proto-oncogene. Okay, so oncogenes and tumor suppressors. Chromosomal defects. That's another thing. Yeah, chromosomes. Chromosomes we'll talk about more when we do mitosis, meiosis, and that kind of stuff, tRNA we talked about. We talked about translation. Initiation factors, beginning translation. rRNA is ribosomal RNA. Oh, sRNA. Oh, there's some like little small other types of RNA. Oh yeah, there's some little small types of other RNA. This is very, very like niche. It's not super where is it? Yes, okay. So there's a couple different types of RNA. I don't think you need to know most of. I think you just need to know ribosomal RNA, transfer RNA, and small and mRNA, right? So the mRNA is the one that's going to leave the nucleus and be translated into proteins. The ribosomal RNA is what is giving rise to those ribosomes. The transfer RNA is the one carrying those amino acids and assisting in the translation. Small nuclear RNA is just makes these spliceosomes, which is involved in ribosome production. Small interfering RNA is something that we can use in the lab to interfere with RNA. It binds complementary mRNA and it messes with it. So if you know that there's mRNA for a protein and you don't want that protein, the small interfering RNA will bind to the mRNA and screw it up, prevent it from being translated. MicroRNA, apparently the same thing, functional RNA interference. Same thing. I'm not sure what the difference is between those. Don't worry about these guys. Just know that that they exist, but it's not something huge like the other ones. Okay, so I think that is basically, yeah, I think that's essentially everything that we need to know about DNA synthesis, RNA's role in the central dogma, and the molecular reasons for why these things are the structure that they are and how they function and all this stuff. Any questions before we wrap up? Okay, so I will stop